Welcome to Lecture 4 of Biology 121, Human Biology and Evolution. This lecture is about macroevolution, or where new species come from. We will begin like we normally do with a metacognition activity. So please take a moment to jot down what you already know about these three questions. Number 1, what is a species? Where do new species come from? And if two related groups are growing apart, when do they become separate species? Feel free to pause this video and take as much time as you need to write down everything you already know. Evolution has been observed on a couple of different levels, and we call these the microevolutionary level and the macro level. Microevolution refers to observed genetic changes, and that's what we talked about in Unit 3. So here at the top you see an ancestral DNA sequence. Over time, lineages are accumulating separate mutations, and they become less and less alike over generations. This change in the DNA sequence is microevolution. You may recall that mutations are copying errors during cell division. So when DNA is going into new cells, it gets copied, and if there's a proofreading error, one of the letters, those A's, T's, C's, and G's on the DNA, mutates into some different letter. And mutations that are heritable are those that happen in the cells that produce egg and sperm. So these are the only mutations that get passed down to offspring. Mutations can happen in any cell in the body, but the ones we're talking about in terms of microevolutionary processes are the heritable ones. So the ones that affect eggs if you're a woman, or sperm if you're a guy. A mutation is the only source of new variation. Mutations are rather rare. There are 1 or 2 mutations per 100 million nucleotides each generation. Remember, nucleotides is the word we use to describe those A's, T's, C's, and G's, the letters of DNA. Since the human genome has 3.2 billion nucleotides, and we know the rate at which they mutate just by random chance, all else equal, we expect between about 30 and 60 unique mutations per person. Over generational time, these mutations slowly increase the genetic variation within the population. A mutation that becomes somewhat common, it originally showed up in just one person, but since they passed it down to their descendants, if they had a lot of descendants, it became more and more common over time. We call this a polymorphism. It's a special type of DNA bit that has become kind of frequent. But the scale of frequency we're talking about here is just 1%. So if a polymorphism reaches a frequency where it's in 1% or more of the entire global species, like all humans everywhere, then it graduates from being called a rare mutation to a polymorphism, which is a somewhat common mutation. So examples of polymorphisms that you've heard about are lactase persistence and the sickle cell polymorphism. That sickle cell allele you studied for lab in Unit 3. So these mutations are accumulating in separate lineages, and eventually that can lead those lineages to become so different that scientists would classify them as different species. But here's where it gets slightly more nuanced. Species are not demarcated by mutations. At least there's no strict number of mutations that separates one species from another, although it is true that species tend to have different sets of polymorphisms. So here's a Chihuahua. Oh my God, it's so small and cute. And a Great Dane. Oh my God, it's so big and cute. There's no magic number of differences that would make dogs distinct species. Here, Chihuahuas and Great Danes, they have a lot of genetic differences. They have clearly a lot of physical differences, yet they're the same species. Contrast that with two species like Neanderthals, which are now extinct, and humans. And we had a lot of DNA similarities and a lot of physical similarities, so much so that we know Neanderthals and humans could have babies together, and yet they're classified as different species. The point is that species are not defined based on the number of mutations separating them. That's not a strict line. So we talked about microevolution. This is macroevolution. This is observed in bodies and behavior. Macroevolution, which is defined in terms of bodies and behavior, this is what defines species. Macroevolution is also where new species come from. So where do new species come from? How does this work? It's important when we're talking about species to remember something. We must remember not to confuse reality with how we choose to describe it. Species are an abstraction. This means that species, this concept, describes groups of organisms that are alike to one another, but humans get to decide where to draw that line. So if there is variation, and that variation appears to blend from one species into another, at some point we just have to draw the line and say, on this side is one species and on that side is another. So we're going to talk about different ways, different pieces of logic that can be employed to draw that line. But in reality, life forms blend. They blend over time, evolving from one into the next, and they also blend from place to place, genetically and in physical traits. So here's the definition of species. It's a group of similar organisms that can successfully breed in nature. They do not typically reproduce with organisms outside their group. Here I'm showing African lions and Asian tigers. While we know that lions and tigers can reproduce in zoos, they don't meet in nature because they live on different continents, so they're considered different species. They're never going to meet each other to produce a little liger in nature. Nature keeps them separate. The same evolutionary processes we've discussed within species may generate new ones. Mutations have been occurring for millions of years independently in separate lineages. So here I'm showing a cladogram with my cartoon stars representing different mutations in different lineages. And those separate sets of mutations accumulating can lead to differences that eventually cause the organisms to diverge and to become different species. And the slow accumulation of genetic differences accounts for the blending we see, so closely related forms in space and time. One species could evolve into another. So thinking about that, here is a thought experiment. Imagine standing side by side with your mother holding her hand. You two look similar because you share half of your DNA. Now imagine on your mother's other side is her mother. So I'm showing this multi-generational family here of a grandmother, her daughter, and the granddaughter. Although you may share some traits with your grandmother, she is less like you than your own mom. Extending this logic down your line of ancestors, you may reach a point in this cosmic family reunion where, although looking similar to her own mother and daughter, the individual looks so different from you that you would no longer call her a human. Each generation looks like the previous. However, distant generations look dissimilar. Far enough down the line, you would no longer recognize that individual as a human. And you can observe this in fossils within the human family tree. So at Pima Community College, we have the privilege of having replica fossils of a number of different ancestors to modern day humans. And we could observe these. We could measure how prominent their eyebrow arches come out, or we could measure the shape of the teeth in their mouth, and we could measure their bone structures. And based on similarities in their forms and the size of these different features, we could build a cladogram, a family tree, a phylogeny. These terms I'm using interchangeably, based on the phenotypic or the physical characteristics we observe in these skulls. So here are their pictures on our human family tree. If we're thinking about our thought experiment, these life forms, more ancient ones, tend to blend into the more recent ancestors. But where do you draw the line? When is it a new species? Scientists have come up with different logic rules to apply to defining species, and they're applied in different contexts. So I'm going to teach you two of these species concepts, and we'll talk about why it makes sense to have both at the same time. So the standard species concept is called the biological species concept. And this defines membership in a species where members can mate with each other and produce viable offspring. That contrasts the second species concept, which is the ecological species concept, in which members are adapted to a particular environment so that hybrid offspring that have intermediate traits, they just fare worse than either parent. So here's some examples to make it more clear. Grizzly bears and polar bears, I told you last lecture, are genetically similar. And so they have no problem reproducing. And in nature, their ranges overlap, and they're becoming more overlapping due to global climate change. So in nature, we have observed many growlers, which is a hybrid with a polar bear parent and a grizzly bear parent. And hybrids are perfectly healthy. They can reproduce with each other, or they can reproduce with either parent species. So contrast that with mules. A horse and a donkey can have a baby, and we call that a mule, but mules are always sterile, meaning mules cannot reproduce. So in the horse, donkey, mule example, which species concept separates horses and donkeys? Hopefully you said the biological species concept, because that's the one that says they cannot reproduce to make fertile offspring. So horses and donkeys are different biological species. However, grizzly and polar bears are separated by the ecological species concept. And here I'm showing you pictures. Imagine one of these brownish colored growlers trying to hide in the Arctic where it's snow covered. Well, they would stand out, and that might be a survival disadvantage if their prey can see them coming. So it's thought that growlers are less successful in the environment of grizzly bears and in the environment of polar bears, and so that keeps grizzlies and polars separate species. We call the process that produces new species speciation. One thing that leads to speciation is reproductive isolation between two lineages. So when two populations of the same species get separated and they no longer interbreed, we say there's no gene flow going from the gene pool of one group to the gene pool of the other. There's no migration. So they're truly isolated. We think that over time, this can lead to speciation because different mutations, by chance, are accumulating in these separated lineages. Here's some different types of speciation when two populations get separated. And I want to explain this like a story. So here's story time about chimps versus gorillas. Chimps and gorillas have overlapping ranges. They both live in African rainforests, and yet they don't interbreed. In the wild, there's never been an observed example of a churilla or a garimp. There's no hybrid offspring. They have no interest in one another. And this is despite many similarities. So here's a chimpanzee baby on the left and a gorilla baby on the right. Oh my God, they're so cute. So some physical similarities are that they're both great apes. So they don't have a tail. They're large-bodied apes. They have big brains. These are really smart species of animals. They're knuckle walkers, meaning they walk on all fours. They produce few offspring at a time with a long period of dependency. Behaviorally, they're both intelligent. They're very social creatures. They live in groups. They have a lot of maternal investment. So mom sticks around to raise the baby for years, just like humans, although not quite as long as humans. They can both live in forests and on the ground. They use tools, and there's been observations in the wild of groups of gorillas fighting with other groups of gorillas. Similarly, in chimpanzees, there are different factions of chimps that war with one another. But they do have some physical differences. So here's a chimpanzee versus a gorilla skeleton. Gorillas tend to be larger. Their teeth shape is different. And they have behavioral differences as well. So chimps have been observed to use tools to crack nuts open, to fish for termites. So over here on the right, what's happening is there is an ape using a stick that the ape has removed all of the leaves from, and it can put this down very carefully into a termite mound. You can see that on the bottom, and when she pulls it up very carefully, termites will have crawled on to the stick, and she can clean it off in her teeth like removing pieces of meat from a shish kebab. Gorillas have never been observed to do these behaviors. Another example that only happens in chimps is hunting. Chimps hunt, like this example is a red colobus monkey. You can see the chimp on the right caught one, it's dead, and he's gonna eat that monkey. And they hunt them on purpose in teams. It's a very interesting hunting behavior. It's coordinated among multiple chimps. And in chimps, they live in groups with multiple males and females together, whereas in gorillas, there's usually a group of one male and a harem of females. So chimp group sizes tend to be a little bit larger, maybe 50 chimps together in an extended family troop. And here's the cool behaviors that gorillas have been witnessed to do. So great apes, they're all muscle, so they sink really quickly and they can't swim. So what this gorilla is doing, you can see in her right hand, she has a stick, and she's putting it in front of her to test the depth of the water before taking a next step. And this is how she's safely crossing the river. Isn't that genius? I mentioned before that gorillas live in smaller groups than chimpanzee. So here is a male with several females, and then he is the father of all the offspring produced. So despite living in the same places and having a lot of similarities in intellect and overlapping behaviors and overlapping morphological characteristics, I mean, not identical, but they're pretty similar. How did the chimpanzee and gorilla lineages diverge? Scientists think that gene flow eventually stopped when differences became too great. So in this example, time is flowing from the top down. And so you can see there's just one channel where all of the individuals are in one group. They're equally likely to reproduce with one another. So this is one population of potentially interbreeding individuals, and the dots are different colors to represent the variation in this one species. Now, over time, due to random chance, mutations start to occur in that gene pool. And if individuals that are similar to each other choose one another for mates, over time, even though they're in the overlapping geographical space, so they have access to all of these other potential mating partners, they become more and more distinct if they're more likely to mate with individuals similar to themselves. And so what we see is in these two diverging channels towards the bottom of this image, the dots start to specialize. There's more orange dots on the channel to the left and more bluish, purplish dots to the channel to the right. And while there may be overlapping gene flow at first, over many generations, they become so distinct in their behaviors and in the way they look that they no longer recognize each other as suitable mates. We call this type of speciation sympatric speciation because the groups are overlapping. It's just something happens and they quit reproducing together. And the two lineages become separate species, and we think that's what happened with chimpanzees and gorillas. They no longer recognize each other as mates. Here's the second story. And this is a story of chimpanzees versus bonobos. Here's a picture of a chimp on the left and a bonobo, which looks very similar to a chimp, on the right. And in the lecture videos for this week, I've included this one. There's a link to it directly from D2L that explains what bonobos are, if you've never heard of them before. They have distinct behaviors from chimpanzees, and they look a little different too, but it's not as immediately apparent as the differences between gorillas and chimps. So in chimps, males are dominant, but in bonobos, females are dominant and run the society. Chimps tend to be very aggressive and coercive, whereas in bonobos, the females form coalitions. They use sex, actually, to cement social bonds, and society is rather peaceful. Chimps, however, have been known to attack and murder each other. In nature, the ranges of chimps and bonobos do not overlap. Historically, and I'm talking in ancient times, they used to be one population, but then the Congo River emerged. From geological evidence, we know this happened about 2 million years ago. And now bonobos exist south of the Congo River and chimps exist north of the Congo River. And remember, I told you great apes can't swim. and this is a large river. So we think they were separated, and these two lineages could no longer get from one side of the river to the other, and that's how they became distinct. We call this allopatric speciation when there is a geographic boundary, like a river or a mountain, maybe the Grand Canyon. That would be a clear geographic boundary, or an ocean, that causes a single ancestral group to diverge into two separate groups. Microevolution is happening independently in each of these groups, so each one is randomly accumulating mutation, but because mutations occur randomly, it's going to be different mixes of mutations accumulating in these separate groups. I include this because it has been a crowd pleaser in my former classes. This is a children's book called Little Changes by Tiffany Taylor, and it describes in a really cute fictional example allopatric speciation in a species of rinckadinks. So there's habitat and dietary specialization in these rinckadink creatures. Here I've given you the link. There's also a link in D2L where you can actually go, and there's a free e-copy of the book. So I don't know if you have little ones at home, you could read it to them or read it to yourself while you're studying for the exam. It's real cute. All right, so you've learned about two types of speciation. Sympatric speciation, where overlapping groups, just for whatever reason, quit reproducing together. And the example I gave you was chimps versus gorillas. The other type is allopatric speciation, which is due to a geographic isolation that prevents interbreeding. And we think this led to chimps versus bonobos, and also that's what happened in the rinckadink example in that children's book. So let this test your knowledge. Which type of speciation applies? Let's do the upper image first. There's an original population. A polymorphism occurs. Over time, there's some reproductive isolation within the same population, and then there's a blue and a green species. Which type of speciation is this? I hope you said sympatric. So the top image is sympatric speciation. Let's contrast that with the bottom image where there's an original population and then there's a barrier that goes up. Perhaps somebody built a wall separating one half of the range from the other. Over generational time, due to reproductive isolation and independent mutations accumulating on either side of the wall, we have a blue and a green species. That's allopatric speciation, due to that geographic barrier preventing interbreeding. So how frequently do new species pop up? What is the rate of speciation in nature? And the answer is it depends. The rate of speciation really depends on how stable the environment is. If the environment is super stable, meaning environmental factors don't change a lot over time. So the climate stays the same type of food exists for many generations. Nesting sites are of the same quality many thousands of years in a row. So everything's kind of the same. Then speciation is rare. But speciation is rapid if the environment changes frequently, it's constantly adjusting, and then the animals that live there have to adjust to suit. Or when the environment contains many different niches, and a niche is an aspect of the environment that an organism could specialize on. So it involves the places where you live and raise your children and the foods that you eat. It allows speciation as individuals develop mutations that give them a competitive advantage in the very specialized, we call it niche space. So if you have a competitive advantage that you can get to all of the seeds of a particular plant fastest and eat them all before the other animals can get to them, then you're going to pass on that trait to your descendants, and they might become their own species. Speciation is also rapid if an environment was previously uninhabited. So when there's zero competition, individuals can specialize on different things in this wide open environment, and that causes speciation to happen quickly over time. And one example of that is called adaptive radiation, when many species arise in a brief amount of time. An example of this is the rise of mammals after the large dinosaurs went extinct. So what you see in this image is something that to me looks like a T-Rex, and then the famous meteorite you can see coming down in the distance. So everything's peachy, but once that meteorite hits, we know that the large dinosaurs went extinct, and so they were no longer eating food, and they were no longer controlling parts of the environment where these tiny little mammals may have previously liked to live. So once the dinosaurs were eliminated, the mammals that survived the meteorite impact were able to invade all of these new niches in the habitat. Without competition from the dinosaurs, they could eat more different types of food. Without competition from the dinosaurs, they could live wherever they wanted. And so what we see right after the dinosaurs go extinct is this explosion in the number of different types of mammals in the fossil record. So all at once, there seemed to be big differences in like the body plan and the sizes and the shapes of teeth, and just a lot of different mammals popping up in a short amount of time. So adaptive radiation, that's one example of that. Another example is the Galapagos Islands, which were originally uninhabited. And then we think a mainland group of birds arrived. And because there was no competition there, they could specialize into all these unique aspects of the environment. And very quickly over time, they developed into separate species of birds, each specializing on its own type of food. If you're interested in Galapagos finches at all, there's this really cool book. It won the Pulitzer Prize. It's called The Beak of the Finch. It explains Darwin's theory of evolution by natural selection in great detail, and I like it because it also explains who the scientists were and kind of makes it a story about their lives and their careers, and unveils the process of science that went into understanding Galapagos finch adaptive radiation. So I invite you to nerd out on that book in your free time if you're into it. A third example of adaptive radiation happened on the Hawaiian Islands. If you've ever heard of Hawaiian honey creepers, here's some images. They're this beautiful group of birds that have beautiful colored feathers, differently shaped beaks, because similar to the Galapagos finches, an original group of birds was blown onto a previously vacant island. It didn't have any birds. And so these speciated really quickly to specialize in different plants found on the islands. And you can see based on what they eat, their beaks became all of these interesting shapes. So I said that the rate of speciation depends on the environment, and that speciation is rapid in a number of different scenarios. And another one is that speciation can happen quickly if the founder population on a previously uninhabited patch of earth, like an island, if it happened to have had a weird mix of polymorphisms, we call this a founder effect. where every individual on the island over time usually has the same polymorphism. So it comes to fixation, meaning everybody there has the same bit of DNA. And it's because it was prevalent in the original founding population. So that's founder effect. And I include a Mr. Potato Head video here on YouTube if you want to learn about founder effect and something called genetic drift. Genetic drift, you can think about it as the survival of the lucky. Random changes can occur in the gene pool, so the frequency of these alleles of DNA can change due to chance, and this is more likely to happen in small populations, and it's when an example would be like a natural disaster strikes and it happens to flood part of an island, and all the individuals, just by bad luck on that half of the island, were wiped out. So once the flood waters recede, the island is vacant for habitation again, and it's the survivors that populate the next generation. Well, if they all happen to have some weird genetic polymorphism, now the island is filled with individuals that have that polymorphism, just because the ones, so many individuals got wiped out by the flood. So by chance, an allele could become really common, or it could be wiped out. The point about genetic drift, though, is that it's always reducing genetic diversity. Meaning, after genetic drift, all individuals are more alike than they used to be. Okay, so here is our after metacognition moment. Please take a moment to record reflections on your learning journey during this lecture. So, was I able to introduce any more details to your previous understanding of what is a species, where do species come from, and if two related groups are growing apart, when is it that they become separate species? Thanks for listening. Bye.